Over recent decades, our knowledge of dietary fibre and starch has expanded. This has led to a new physiological-based definition of dietary fibre, and a new methodology has been developed, leading to AOAC method 2017.16 and ICC method 185 for total dietary fibre. In parallel, accurate, updated methodology for the measurement of resistant starch has been developed. This rapid resistant starch assay kit can be used to accurately measure resistant starch in various sample types throughout industries such as cereals, food and beverage, and animal feeds. This video tutorial will demonstrate the use of the kit with 2 cereal samples and the kit control using the manual spectrophotometer format. The kit contains sufficient reagents for 100 manual assays and is supplied with a detailed data booklet. The principle of the rapid resistant starch assay is shown in these slides. Starch samples are incubated with saturating levels of purified pancreatic alpha amylase and amyloglucosidase for 4 hours at 37 degrees Celsius in a shaking water bath in linear motion. Digestible, non-resistant starch is solubilized during this time and hydrolyzed to D-glucose. Any remaining starch is classified as resistant starch. After 4 hours, the reaction is terminated by adding ethanol or IMS, and the resistant starch is recovered by centrifugation. Once washed, the resistant starch in the pellet is then dissolved in ice-cold 1.7 molar NaOH with vigorous stirring. Upon neutralization with acetate buffer, the dissolved resistant starch is hydrolyzed to D-glucose with amyloglucosidase. D-glucose is quantitatively measured with glucose oxidase peroxidase GoPod reagent. The amount of D-glucose measured is stoichiometric with the amount of resistant starch in the sample. Prior to sample analysis, prepare the required additional reagents as described in the kit data booklet. Subsequently, once all of the additional reagents have been made, the kit components should be prepared, and once prepared, they are ready for use in the manual assay procedure. Use the contents of bottles 2, 5, and 6 as supplied. Also, prepare a solution of diluted amyloglucosidase as described. Immediately prior to use, add 0.1 grams of the contents of bottle 1, pancreatic alpha amylase and amyloglucosidase, to 5 milliliters of sodium maleate buffer and stir on a vortex for 5 minutes. This solution should be stored on ice during use and should be used within 4 hours of preparation. Alternatively, the pancreatic alpha amylase can be prepared as a relatively stable ammonium sulfate suspension for use over approximately 3 months as follows. Add 0.5 grams of bottle 1 pancreatic alpha amylase and amyloglucosidase to 7 milliliters of cold distilled water in a 16 milliliter polypropylene tube while stirring in a fume hood. Stir for approximately 5 minutes until the enzymes are completely dissolved. Add 3.5 grams of solid ammonium sulfate and dissolve by stirring. Whilst taking into account the stirrer bar, adjust the volume to 10 milliliters with 50% weight per volume ammonium sulfate solution. Cap the tube and store at 4 degrees Celsius. This suspension is stable for up to 3 months. Transfer 1 milliliters of bottle 2 to 30 milliliters of 100 millimolar sodium acetate buffer, pH 4.5, and mix well. Store aliquots of approximately 10 milliliters of the solution in a freezer, labelled as amyloglucosidase at approximately 100 units per ml in sodium acetate buffer, pH 4.5. Dilute the contents of bottle 3 GoPod reagent buffer to 1 litre with distilled water. This is solution 3. This should be used immediately. Dissolve the contents of bottle 4 GoPod reagent enzymes in 20 ml of solution 3. Quantitatively transfer this to the graduated cylinder containing the rest of solution 3. This is the glucose determination GoPod reagent, which is stable for approximately 3 months at 2 to 5 degrees Celsius. Cover this bottle with aluminium foil to protect from light. For longer term storage, divide into approximately 250 milliliter aliquots and store at -10 degrees Celsius. This is stable for greater than 12 months at -10 degrees Celsius and should not undergo more than one freeze-thaw cycle. Accurately weigh approximately 100 milligrams of sample directly into a 13 milliliter polypropylene tube. Gently tap the tube to ensure all of the sample falls to the bottom and record the exact weight. In this demonstration, we are using 2 serial samples in duplicate and the provided kit control in duplicate. Add 3.5 milliliters of sodium maleate buffer pH 6.0. Cap the tubes and mix the contents on a vortex mixer for 5 seconds. Incubate the tubes in a 37 degrees Celsius water bath and allow the tubes to equilibrate to temperature over 5 minutes. After 5 minutes, uncap the tubes and add 0.5 milliliters of the prepared pancreatic alpha amylase and amyloglucosidase solution to the tubes. Alternatively, if using the ammonium sulfate suspension of these enzymes, add 3.8 milliliters of sodium maleate buffer, pH 6. Cap the tubes and incubate at 37 degrees Celsius for 5 minutes. After 5 minutes, uncap the tubes and add 0.2 milliliters of the alpha amylase and amyloglucosidase ammonium sulfate enzyme suspension. Cap the tubes tightly and attach them horizontally aligned in the direction of motion in a shaking water bath. Incubate the tubes at 37 degrees Celsius with continuous shaking at 200 strokes per minute for exactly 4 hours. After exactly 4 hours incubation, remove the tubes from the water bath and quickly remove the caps. Immediately add 4 milliliters of 95% volume per volume ethanol or IMS to each tube. Cap the tubes and mix the tube contents vigorously on a vortex mixer. After mixing, remove the caps and centrifuge the tubes at 4000 RPM for 10 minutes. Immediately after the centrifuge stops, carefully decant the supernatant solution, ensuring that the pellets are not disturbed. This solution should be retained for the measurement of digestible starch. Resuspend the pellet in 2 milliliters of 50% volume per volume ethanol or IMS with vigorous stirring on a vortex mixer. Add a further 6 milliliters of 50% volume per volume ethanol or IMS. Cap the tubes and mix thoroughly by inversion. Remove the lids and centrifuge at 4000 RPM for 10 minutes. Decant the supernatant solutions as before and repeat the suspension and centrifugation steps once more. Invert the open tubes on absorbent paper to remove any excess liquid, ensuring that the pellet is not dislodged. Combine all of the sample-specific supernatants for the determination of digestible starch. Add a 6 by 12 mm stirrer bar to each tube. Add 2 ml of 1.7 molar NaOH to each tube and dissolve the resistant starch. Resuspend the pellets by stirring for approximately 20 minutes in an ice water bath over a magnetic stirrer. It is important not to use a vortex mixer as the sample may emulsify. Add 8 ml of 1 molar sodium acetate buffer pH 3.8 to each stirring tube and mix thoroughly. Remove the stirring tubes and immediately add 0.1 ml of bottle 2 amyloglucosidase at 3,300 units per ml. Cap the tubes, mix thoroughly on a vortex mixer. Incubate the tubes at 50 degrees Celsius for 30 minutes. With intermittent mixing on a vortex mixer. For samples containing less than 10% resistant starch, no dilution is required. Therefore, remove stirrer bars, vortex, and directly centrifuge the tubes at 4000 RPM for 10 minutes. For samples containing greater than 10% resistant starch, quantitatively transfer the contents of the tube to a 100 ml volumetric flask using a water wash bottle. Adjust to 100 ml with distilled water and mix well. Centrifuge an aliquot of the solution at 4000 RPM for 10 minutes. Transfer 0.1 ml aliquots in duplicate of the supernatants from the centrifuge samples into labelled glass test tubes. Prepare the D-glucose standards by adding 0.1 ml of D-glucose standard solution, bottle 5, to tubes in quadruplicate. Prepare the reagent blank solutions by adding 0.1 ml of 100 millimolar sodium acetate buffer, pH 4.5, to tubes in duplicate. Add 3 ml of GoPod reagent to all tubes and incubate at 50 degrees Celsius for 20 minutes. After the 20-minute incubation period, measure the absorbance of each solution at 510 nanometers against the reagent blank. Use the absorbances to calculate the content of resistant starch. Combine the supernatant solutions achieved in the hydrolysis of digestible starch section and adjust the volume to 100 ml with distilled water in a volumetric flask. Mix the contents well. Transfer an aliquot of these solutions to labelled microfuge tubes for easy pipetting. Transfer 0.1 ml aliquots of the sample solutions to the bottom of duplicate 16 by 100 mm tubes and add 0.1 ml of the diluted amyloglucosidase at 100 units per ml. Incubate at 50 degrees Celsius for 30 minutes to hydrolyse remaining traces of maltose in the samples. During the 30-minute incubation period, prepare the standards and blanks for glucose determination. Glucose standard solutions are prepared by mixing 0.1 ml of glucose at 1 mg per ml with 0.1 ml of 100 mmol sodium acetate buffer pH 4.5 to tubes in quadruplicate. Prepare reagent blanks by adding 0.2 ml of 100 mmol sodium acetate buffer pH 4.5 to tubes in duplicate. After the 30-minute incubation period, remove tubes from the water bath. Add 3 ml of GoPod reagent to all tubes. And incubate the tubes for a further 20 minutes at 50 degrees Celsius. Measure the absorbance at 510 nanometers against a reagent blank. Use the absorbances to calculate the content of digestible, non-resistant starch. The absorbance readings of the sample and the blank reactions are used to calculate the resistant starch percentage on an as-is or dry weight basis in the test samples. When performing this test on the megaquant wave spectrophotometer, the absorbance mode feature must be used. The results will be printed via the onboard printer, or the data can be exported to a computer using the SF Capture software. Please see our Megaquant Wave video for further details. In absorbance mode, the results output are raw absorbance values for both blank and samples. Therefore, the calculations of resistant starch, digestible starch, and subsequently total starch content can be performed manually as described in the calculation section of the kit booklet. Megazyme has also developed specific Excel-based MegaCalc applications for each Megazyme kit to allow quick and easy results analysis. Results can be analysed using the Megacalc application specific to this rapid resistant starch assay kit, which is available to download free of charge from the Megazyme website. Results can be calculated on an as-is basis, or moisture content of the sample can be allowed for. The Megacalc spreadsheet provides full instructions for use. Open the Megacalc worksheet and input the following: sample details, absorbance readings for the D-glucose standards. For each sample, input the sample identifier. Input the sample weight used. Alter the extract volume if a volume other than the default 10.3 milliliters has been used. If the samples have greater than 10% resistant starch, the extract volume will be 100 milliliters. For each sample, input the absorbance values recorded in duplicate. Alter the sample volume if a volume other than the default 0.1 milliliters is used. If dilution of the sample has been performed prior to the GoPod assay, then input the dilution factor used. If no further dilution was performed, the dilution factor is 1. When all of the data has been entered, the percentage of resistant starch in the sample is automatically calculated and given as grams per 100 gram on an as-is basis. If the moisture content of the samples is known, input the moisture content percentage. When this additional data is entered, the concentration of resistant starch is automatically calculated and given as grams per 100 gram on a dry weight basis. The corresponding digestible starch fractions of the samples can be also calculated in this manner. The sum of the resistant starch plus the digestible starch gives the total starch content of the samples. For further information, the relevance and measurement of resistant starch, digestible starch, and available carbohydrates is discussed in detail in a paper by McCleary et al.